হ্যালো বন্ধুরা, আমি হারুন। এটা তোমাদের ইউটিউব চ্যানেল হারুন বায়োজোন। আজকে আমি এই ভিডিওটি নিয়ে এসেছি ক্লাস 12-এর চ্যাপ্টার 11 বায়োটেকনোলজি প্রিন্সিপাল অ্যান্ড প্রসেস। এই ভিডিওতে আমি এই অধ্যায়ের 60টি খুব ভেরি ইম্পর্ট্যান্ট কোয়েশ্চেন তোমাদের সঙ্গে পুরো ডিটেলসে আলোচনা করব, যেগুলো তোমাদের বিভিন্ন মেডিকেল সহ বিভিন্ন পরীক্ষার জন্য খুবই গুরুত্বপূর্ণ। তো মিস না করে এই ভিডিওটি দেখো, চলো দেরি না করে শুরু করা যাক। তাহলে আজকের প্রথম প্রশ্ন হচ্ছে জেনেটিক ইঞ্জিনিয়ারিং। জেনেটিক ইঞ্জিনিয়ারিং ইজ what is genetic engineering? Making artificial gene or hybridization of DNA of one organism to that of others, or production of alcohol by using microorganisms. Making artificial limbs, diagnostic instrument and from other source and some other instrumental source like ECG is. So this is a medical engineering, okay, and production of alcohol by using microorganism. This is traditional biotechnology, that use human from long time ago. Traditional biotechnology. But hybridization of DNA of one organism to that of others. This is the correct option. Genetic engineering is the hybridization of DNA of one organism to that of others. Making artificial gene also the part of genetic engineering, but this is not complete answer. The complete answer is hybridization of DNA of one organism to that of others. Next question, construction of recombinant DNA. Construction of recombinant DNA involves, what is recombinant DNA? When the vector DNA and the foreign DNA are ligated by ligase enzyme is called recombinant DNA. Here the question I want to say, construction of recombinant DNA involves cleaving and rejoining of DNA segment with endonuclease harun. Cleaving means cutting, and DNA segment with endonuclease and rejoining them with ligase. The first, this is the foreign DNA and this is vector DNA. Clear? The first use restriction endonuclease to cut the foreign DNA and the vector DNA into a small fragment. Okay, use cleave, here cleave, then again rejoin by enzyme ligase. So cleaving the DNA segment with endonuclease. Endonuclease means restriction endonuclease. Because pure endonuclease cut DNA at any segment, but restriction endonuclease cut DNA at a specific sequence. So here the correct option is B. Next question, genetic engineering is possible. Genetic engineering is possible. Why? Because we can cut DNA at a specific site by endonuclease like DNase 1 do not cut DNA at a specific position. It cut DNA at non-specific position, double stranded DNA, single stranded both. Restriction endonuclease purified from bacteria can be used in vitro. Definitely use. The phenomenon of transduction in bacteria is well understood. Not. We can see DNA by electron microscope. Not. Here the genetic engineering is possible because the DNA is cut at first DNA is cut by a specific sequence. by restriction endonuclease. This is the first step of genetic engineering, because the main purpose of genetic engineering to hybridization the organism or increase the product or produce genetically modified organism. So here first work is the cutting the DNA and joining the DNA, then produce a recombinant DNA. The first restriction endonuclease purified from bacteria can be used in vitro. Next question. Cloning gene is a process. Cloning means duplicate copy, duplication. So here the cloning gene is process where gene is cloned in an animal. Fragment of DNA are transferred from one organism to another, usually carried on a DNA vector. Fragment of DNA cloned in the same organ, not same organism. Here the fragment of DNA is transferred from one organism to other, usually carried on a DNA vector. Okay, this is a DNA vector and this is a foreign DNA particle. Okay, this recombinant DNA transferred to another host and the target organism to change the phenotype of that organism. Next question. Which of the following enzyme is used in genetic engineering? Here translocase, DNase, topoisomerase, restriction endonuclease. The first enzyme, the pioneer of the genetic engineering enzyme is restriction endonuclease. So what is the function of other enzyme? Translocase, transfer functional groove from one to another molecules. DNase, cut both single strand and double strand DNA at any site. Topoisomerase, unwind the supercoiled DNA. Okay, and restriction, cut DNA at a specific site. Cut DNA at a specific sequence. Here the correct option is D. Next question. Which enzyme is used as molecular scissor? Molecular scissor means the enzyme that cut the DNA fragment at a specific palindromic sequence. This is called molecular scissor. So here restriction endonuclease is molecular scissor. DNA ligase is molecular glue. DNA polymerize the DNA, means synthesis of DNA, polymerize the DNA. And DNA guidase is a unwinding of DNA. Next question is blunt end of passenger and vehicle's DNA are joined by DNA ligase, because this is called molecular glue. DNA polymerase used during replication, and DNA polymerase 3 are also used during replication to polymerize the DNA and error correction. RNA polymerase used during transcription to synthesize mRNA. Next question. The technique in which foreign DNA is precipitated over surface of metal particle for passing into the target cell. This is the technique by which the foreign DNA transfer into the host cell. Micro injection. This is a syringe and through which the foreign DNA is transferred into the, especially in animal cell. Electroporation. Electroporation means use electric supply to increase the permeability of cell membrane, so the foreign DNA is inserted into the bacterial cell, host cell. Particles gun. This is a method where the foreign DNA is transferred into the host cell, especially in case of plant cell, because in case of plant cell there is cell wall present. So the gene is first, the foreign gene is covered by a tungsten or gold particle, then it transfer to the host cell forcefully by gene gun method or some of the particle bombardment. Okay, chemical mediated gene transfer. Chemical mediated gene transfer is also a process through which the gene transfer into the host cell. So here the foreign DNA is precipitated over surface of metal particles. Metal means gold and particle, gold tungsten. So here the foreign DNA is covered with gold or tungsten, then transferred into the host cell by particle gun or gene gun method. Next question is, restriction enzymes present in several microorganisms cut foreign DNA at a specific site, and destroy them. The enzyme do not destroy the cellular DNA. Here the function of restriction endonuclease in bacteria, when the bacteria is affected by, when the bacteria is attacked by bacteriophage, this restriction endonuclease has two functions. First, this enzyme methylated the own DNA. This enzyme methylated the own DNA, own bacterial DNA, and second, it fragmented the foreign DNA. First, so the cellular DNA does not have the specific site. B, the susceptible specific sites are masked by proteins. The restriction enzyme susceptible sites are modified by cellular enzyme. The susceptible sites are modified by cellular enzyme. The restriction enzyme and DNA occupy different component. So here first function is, first the enzyme has two functions. One, it in the presence of methylase, they methylated his own DNA. So the restriction enzyme susceptible site are modified by catalyzed by particular enzyme methylase. The cellular DNA does not have the specific site. The susceptible specific sites are masked by proteins. The restriction enzyme susceptible sites are modified by cellular enzyme. Which enzyme? The methylase enzyme. Okay, and the number two function is cut the viral DNA. Two function of restriction. So here the C is the correct option. Next question is, plasmids are extra chromosomal circular DNA. Suppose this is bacteria and this is the bacterial own DNA and this is the plasmid DNA. So plasmid is extra chromosomal DNA that are independently replicate. So here, which have their own point of replication and can replicate independently. Which have their own point of replication but cannot replicate. This is false. Which do not have their own point of replication and cannot. This is false. This is false. So here the correct option is, because DNA, plasmid DNA is extra chromosomal DNA that are autonomously replicate, and contain very few number of gene, especially it protect the host or E. coli from different antibiotic. Okay. Next question is GAA-TTC is the recognition site for which of the following restriction endonuclease? GAA-TTC CTT-AAG is the recognition site for EcoR1. Here the restriction site occur between G and A. Clear? In case of HindIII, the recognition sequence is AAGCTT 5' to 3' and 3' TTCGA 5'. Here also the recognition site or cutting site is AA and A. So in both of the case the sticky end is developed. But in case of BAM H1, here GGATCC, so CCTAGG. This is BAM1 sequence. Here the cutting site is between G and G, so sticky end are forms. But in case of HAE3, here the 5' GGCC CCGG 5'. So here the blunt end is formed, or GGCCCCGG. This is blunt end. But in case of EcoR1, G C T T A G. This is sequence called sticky end. So here the correct option is B. Next question. Paleontologist means evolutionary biologist, unearthed a human skull during excavation. When excavate, they found a human skull with a small fragment of scalp. Okay, tissue. What is still attached to it? Only little DNA could be extracted. Only little amount of DNA they extracted from the excavation skull. If the gene of ancient man needed to be analyzed, the best way to getting sufficient amount of DNA. So a small amount of DNA is amplify into large amount by which PCR technique, polymerase chain reaction. The subject the DNA not gel electrophoresis separate the DNA on the basis of their size. So not answer. Treating the DNA restriction endonuclease cut the DNA at a specific site. Answer. Hybridizing the DNA with DNA probe, not. DNA probe is small DNA fragment. DNA probe used to identify the unknown DNA sequence. This is also not. Subject the DNA to polymerase chain reaction. They are PCR technique. Here use TAC polymer, Thermus aquaticus bacteria produce this polymerase. And Thermus aquaticus bacteria produce this Taq polymerase. Clear? And Taq polymerase, in the presence of this Taq polymerase, the DNA are amplified into many copies. Next question is, genetically modified food can be harmful because it can cause allergic and toxicity. Maybe sometimes they cause toxicity and allergy. Incorporation of antibiotic resistance in human being, disturbance in metabolism, and due to enzymes for restriction, antibiotic resistance, all of this. So genetically modified food may be harmful because in case of genetic modified food, the normal wild type genetics is changed. So different type of toxic proteins may be produced that cause allergic substance, sometimes toxicity, sometimes metabolism disorders. Okay, and just antibiotic resistance, some antibiotic resistance also. So here the correct option is all of these. Next question is, which one of the following is correct? Correct statement. Bt in Bt cotton indicate that it is a genetically modified organism. Bt in Bt cotton. Bt means Bacillus thuringiensis. Bacillus thuringiensis. This is a gram positive, gram negative bacteria from which the Bt gene isolated. Then be somatic hybridization involved. Somatic hybridization means two somatic cell are hybrid. Example, protoplast fusion. Somatic hybridization involves fusion of two complete plant, not complete plant cell, because it fusion of two protoplast. Protoplast is a cell without cell wall. So no complete cell wall. The anticoagulant hirudin is being produced from transgenic Brassica napus seeds. Anticoagulant hirudin is produced. Hirudin actually from leech. Hirudin gene are isolated. These genes are introduced into the Brassica napus. So when we eat the oil from Brassica napus, it help to help as anticoagulant in our body. Okay, so this is the correct option. Flat slat variety of tomato has enhanced the production of flat slat tomato do not enhance the production of ethylene. Flat slat tomato increase the shelf life. Increase the shelf life of tomato by inhibiting the enzyme beta galacturonidase that stimulate fruit ripening. So the correct option is the Brassica napus produce hirudin gene. These are anticoagulant. Next question. The Bt toxin is not toxic to human but toxic to bollworm. But toxic to why? Toxic to bollworm, or that eat the leaf of the tobacco plant. The pro-Bt toxin activation requires temperature, not temperature. Bt toxin recognize only insect-specific target. This is also not right. Bt toxin formation from pre-Bt toxin require pH lower than that present in human stomach. Conversion of pro-Bt toxin to Bt toxin takes place only in high alkaline condition. So in case of human being, it is not effective. In case of human being, it is not effective because human gut is acidic in nature. Because human gut is acidic in nature, but Bt toxin require alkaline pH. Clear? So here the correct option is conversion of pro-Bt toxin to Bt toxin takes place only in highly alkaline condition. Okay, that this condition are occur in the stomach of bollworm. So here they produce a pore. Okay, Bt toxin produce a cryprotein in alkaline condition, cryprotein formation a pore in the small intestine. So the bollworm is die. Next question is, which of the following is correctly matches? Agrobacterium tumefaciens that produce tumor, crown gall disease. Where? In dicot plant. Thermus aquaticus that produce Taq polymerase used in PCR. Bt gene produced from Bacillus thuringiensis. PBR322 is a vector which full form is plasmid of Bolivar and Rodriguez. Okay, Bolivar and Rodriguez. Ligase is the molecular glue. Molecular structure is a restriction endonuclease. The correct option is Agrobacterium tumefaciens that cause tumor or crown gall disease, especially dicot plant. Match the column 1 with column 2. First, Riopro. What is Riopro? Riopro is a blood thinner. That prevent blood clot. So Riopro is the prevention blood clot. These are 4. 5. DNase. DNase is an enzyme that DNase actually here recombinant human DNase. DNase means recombinant human DNase. This is treatment of cystic fibrosis. Cystic fibrosis. What happens in cystic fibrosis? Because in case of cystic fibrosis, cough, repeat lungs infection, fatty stool, etc. symptom of the cystic fibrosis. Cystic fibrosis is a autosomal recessive disorder. This recombinant age human DNase, what they do? They thinner the mucus. This cough is produced by some microorganism. This microorganism DNA is cleaved by DNase H. So the cough or mucus become thinner. DNA are used treatment of cystic fibrosis. Interferon alpha is used. Interferon alpha is used cytokine produce innate immunity against hepatitis C. Okay, এবং আর asparginase is treatment against lymphoma virus, lymphoma cancer. And OKT3 is a immunosuppressant drug that is used to prevent the graft rejection or suppose reversal of acute kidney transplantation. So here A is 5, B is 4, DNA is treatment cystic fibrosis, C interferon alpha is hepatitis C, D asparginase is treatment of cancer, and E OKT3 is a reversal of acute kidney transplant rejection. It is a immunosuppressant. Okay. Here A is the correct option. Next question is, find the incorrect statement. Gene therapy is a genetic engineering technique used to treat disease at molecular level by replacing defective gene with normal genes. Right option. It is a correct on. Calcitonin is a medically used recombinant protein. Calcitonin produced from C cell of thyroid maintain blood calcium level. Okay, and britoxin is a biodegradable insecticide obtained from Bacillus thuringiensis. This is also right on. And trichoderma is biocontrol agent for fungal disease of plant. All right. So here the incorrect option is B. Next question. Match list 1 with list 2 and select the correct option. First, Bacillus thuringiensis. The Bacillus thuringiensis bacteria produce insecticidal gene, that is britoxin. Rhizobium meliloti incorporation of neet gene. Neet genes mean nitrogen fixation gene. E. coli bacteria producing of human insulin. And Pseudomonas putida scavenger of oil spills. Okay, Pseudomonas putida break down the hydrocarbon when oil are spilled in the ocean water. Trichoderma is a fungus that produce chitinase. Here A is the 4. Here A is 4. Bacillus thuringiensis produces britoxin. B Rhizobium meliloti neet gene, and C E. coli production of human insulin. D Pseudomonas putida oil spill, and E is 1. Here the correct option is C. Next question is, some of the steps involved in the production of humulin. Humulin means human insulin. Human insulin is called humulin. First proteins are produced by recombinant DNA technology. Synthesis of gene. So here the sequentially arrange the following process. First here synthesis of gene DNA from human insulin artificially, culturing recombinant E. coli in bioreactor, purification of humulin, insertion of human insulin gene into the plasmid, introduction of recombinant plasmid into the E. coli, extraction of recombinant gene product from the E. coli. The first synthesis of gene from human insulin artificially. First work is 1. Then Then insertion of human insulin gene into plasmid. 4. Then culturing recombinant E. coli in bioreactor. 2. Not culturing. First, synthesis of gene DNA from human insulin artificially. Next next insertion of human insulin gene into the plasmid. Okay. Then 4. Then 5. Introduction of recombinant plasmid into the E. coli. Then 5. After 5, culturing recombinant DNA recombinant E. coli into bioreactor. 2. Then then extraction of recombinant gene product from the E. coli. 6. And then purification of humulin. 3. C is the correct option. Next question is, most widely used bioweapons. Bioweapons means the bacillus used as the weapon for killing the human beings. Here Bacillus anthracis is used in food to kill the human being. Pseudomonas fetida, this is oil spills bacteria, and Bacillus subtilis is used as probiotics. Then next question is, which of the following statement correct regarding genetical engineering approval committee? What is the function of genetically engineering approval committee? It makes decision regarding the validity of GM crops. The GM crop is valid or invalid, because or it is helpful or harmful to human being. They research. It ensures the safety of introducing GM organism for public service. Genetically modification of organism can have unpredictable results when such organisms are introduced into the ecosystem. Therefore, the Indian government has set up organization like Genetically Engineering Approval Committee. Here all of the above are the correct answer. Gel electrophoresis is used for construction of recombinant DNA by joining with gel electrophoresis is a gel. This gel is agar gels and collect from the seaweeds Gelidium, gracilaria and Zygartina. Here some oils are present. This is cathode, this is anode. Okay, so here anode means positively charged, this is negatively charged, electric supply. Okay, these well are filled with DNA fragment. Then when the electric supply is done, DNA is negatively charged, so DNA move towards the positive direction. So here the gene DNA So here the DNA is separated from large to small. Because DNA is negatively charged, so move from negative to positive or cathode to anode on the basis of size. The small size move more distance, but large size do not move more distance than small size. Isolation of DNA molecule, cutting of DNA into fragments, separation of DNA fragment according to their size. So here D is the correct option. Next question, which of the following is a case of wrong matching? Somatic hybridization. The somatic hybridization means fusion of two protoplast cell or somatic cell. Vector DNA, site for tRNA. Vector DNA is not tRNA synthesis, it is a carrier of foreign DNA. Site for tRNA synthesis is nucleus. Micropropagation in vitro production of plants in large numbers. This is the right option. callus, unorganized mass of cell produced in tissue culture. This is also right. Somatic and fusion of two diverse cell. This is the right answer. So here the wrong answer is this one, B. Vector DNA is a carrier of foreign DNA transfer, or site of DNA synthesis is nucleus. Next question. The colonies of recombinant bacteria appear white in contrast to blue colonies of non-recombinant bacteria, because here the question want to say how the recombinant DNA is separate from non-recombinant DNA on the basis of color production. So here first you have to know that this is the vector, and in this region, suppose this is beta galactosidase gene, galactosidase. This is beta galactosidase gene. Clear? Okay. Here some foreign DNA are inserted into this region. Normally this beta galactosidase produce blue in color. Okay, blue color. But when some foreign DNA inserted inside this beta galactosidase, the beta galactosidase become inactive. Okay. When inactive, no color is produced. So in case of recombinant DNA, so when inserted means when foreign DNA insert in the beta galactosidase region, these do not show the produce color, because this DNA become recombinant. But in case of non-recombinant DNA, the beta galactosidase become remain active, so it produce blue in color. So here insertion and activation of alpha galactosidase, not alpha galactosidase, it is beta galactosidase. Inactivation of glycosidase, not glycoside. Glycosidase means produce carbohydrate to glucose production enzyme. This is also not answer. Non-recombinant bacteria containing beta galactosidase, because here you can see the beta galactosidase is present when the foreign DNA do not insert into the beta galactosidase gene. So the non-recombinant bacteria containing beta galactosidase, they contain beta galactosidase. Insertion and activation of alpha, not alpha, this is beta. The correct option is C. Which of the following is not correctly matched for the organism and its cell wall degrading enzyme? The algae is a plant. The plant means here cellulase, not methylase. Fungi is a chitinase, and bacteria is lysozyme, because bacterial cell wall is degraded by lysozyme. Plant cell wall, cellulose containing, so cellulase enzyme are used. In case of algae, there are different type of enzymes, okay, are produced. So here the wrong answer is algae. Next question. What is anti-sense technology? Anti-sense, sense means coding strand, or anti means against coding strand. So when a piece of RNA that is complementary in sequence is used to stop expression of a specific gene. RNA polymerase producing DNA, a cell displaying foreign antigen used for synthesis of antigen, production of somaclonal variant. Here the A is the correct option. So it is properly explained with some diagram. Suppose this is antisense oligonucleotide, okay, and this is small interfering RNA. Clear? As oligonucleotide has RNase H1. RNase H1 means it is the enzyme that break down the mRNA. So RNA H once here break down the mRNA, so mRNA become silence. Okay. Suppose this is a foreign DNA or DNA from some Medullary incognita nematode that called hair disease. The S oligonucleotide is a small fragment bind with some mRNA and degrade it due to RNA activity. Some small interfering RNA that antisense that produce antisense strand with the help of RISC. RISC means RNA independent silencing complex. RISC RNA independence silencing complex. RNA independent silencing complex that silence the mRNA expression and degree of mRNA. So the functional mRNA do not express. Functional means the mRNA from some pathogens do not express itself because it degraded by the small interfering RNA and oligonucleotide. So here correct option is when a piece of the RNA that is complementary to sequence is used to stop expression of a specific gene. Next question is main objective or production use of herbicide resistant GM crop. Herbicide or weedicide resistant GM crop. The objective of production of herbicide resistant GM crop because encourage eco-friendly herbicide. There is no eco-friendly herbicide. Reduce herbicide accumulation in food articles for health safety. Why recombinant DNA technology produce herbicide resistance genetically modified plant? Why they produce? Because reduce herbicide because if we use herbicide, the plant absorb it. When we take this plant or eat this plant, this herbicide if the herbicide is non-degradable, it show biomagnification. So from this we can use herbicide resistance on plants. Okay. Reduce herbicide accumulation in foods articles for health safety. Elimination of weeds from the field without the use of manual labor. Because elimination of weeds used some weedicide and elimination of weeds from the field without the use of herbicide, not with the use. So here the correct option is reduce herbicide accumulation in food articles for health safety. Transgenic plants are the ones. Which one is transgenic? Generated by introducing foreign DNA into a cell and regenerating a plant from the cell. Produced after protoplast fusion in artificial medium. Growing in artificial medium after hybridization in the field. Production by somatic embryo in artificial medium. So here the correct transgenic plant means gene transfer within the plant. So change the plant's phenotypes. So generated by introducing foreign DNA into a cell and replicating in plant from that cell. These are transgenic plants. Next, read the following 4 statements and certain mistakes in two of them. The first transgenic buffalo, not buffalo, this is cow, Rosie, produce milk which has human alpha lactalbumin enriched because this lactalbumin is more efficient than human milk. Restriction enzymes are used in isolation of DNA from other macromolecules. Not restriction enzyme. What is the function of restriction enzyme? Cut the DNA at a specific site into a small fragment. This is wrong answer. This is also wrong answer. Downstream processing is one of the steps of recombinant. Downstream processing is the one of the steps of recombinant DNA technology to process and purify the product. The disarmed pathogen vector are also used in transfer of recombinant host. Definitely, disarmed recombinants, the bacteria or virus that cause specific pathogen to the host. If this type of bacteria is isolated and disarmed, they can use as a vector. So here the wrong answer is D, A and B. Consumption of which one of the following foods can prevent the kind of blindness? The blindness means vitamin A. Vitamin A. The vitamin A is in the cultivate. Here, blindness means golden eyes. We are golden beta carotene gene. Or beta carotene gene produce vitamin A. Vitamin A. Canola seeds oil produce vegetable oil. Vegetable oil. BT brinjal insecticide. Flat tomato, where they increase the shelf life of the tomato. Inactivate beta galactoronidase. Next question is, tobacco plant resistance to nematode have been developed by the introduction of DNA that produce both sense and antisense RNA. Okay, antisense RNA we see before. The first human hormone produced by recombinant DNA is insulin. In Bt cotton, the Bt toxin present in plant tissue as a protoxin is converted into active toxin. Actually, what happened? Bt gene. Bollworm gut, alkaline condition produce cryoprotein. Hello? Action of gut microorganism, not. Presence of conversion of factor in insect gut, not. Alkaline pH of insect. Alkaline pH of insect gut is a main cause to activate this Bt gene, but in case of human there is acidic gut, so no harmful effect. Which kind of therapy was given in 1990 to 4 years old girl with adenosine deaminase? It is called SCID, severe combined immunodeficiency. So here, which kind of therapy? Here, gene therapy is used. This name is SCID because adenosine deaminase, due to inactivation of adenosine deaminase, there is no production of lymphocyte. If no lymphocyte produce, no B cell and T cell are produced, so no immune system will occur. And chemotherapy, immunotherapy and radiotherapy are used against cancer. Okay, against cancer. Primers. What is primer? Primer is small nucleotide sequence, about 10 to 18 nucleotide that complementary to the DNA sequence. Small chemically synthesized oligonucleotides of about 10 to 18 nucleotides that are complementary to the region of template DNA. This is the correct answer. When restriction enzymes cut the strand of DNA a little away from the center, if the palindromic sites between the same two base opposition means here produce sticky end, means suppose GAATTC 5 prime to 3 prime C here what happened, this is GAATTC, this is on palindromic sequence, to CTTAAG and the sequence. If the cut is occur between G and A, cut occur G and A, so what happened? GAATTC and CTTAAG. Here the option is sticky end. Ampicillin resistance in vector PBCR322 indicate gene responsible for synthesis of or not. Gene conferring resistance against ampicillin. Ampicillin resistance means the gene present in the PBR322 is ampicillin resistant gene. So, in the presence of ampicillin resistant gene, the PBR322 are survive in ampicillin containing medium. Vector, what is vector? Vectors mean carrier. Molecules that degrade nucleic acid, molecules that help in replication, molecules that are able to covalently bond to and carry foreign DNA into cell, molecules that protect host cell from invasion or false. This is the C as correct answer. Vector are very small sized DNA that carry the foreign DNA into the host cell. Agarobacterium tumefaciens that cause crown gall disease, a disease in human that cause loss of sight, not false. The dicot plant, a bacterium that can be used to introduce into DNA into plants. This is the correct answer. B, not fungus, it is bacteria. Key tools to involve in recombinant DNA technologies. So, which tools are used? Restriction endogenous nuclease for cutting the DNA into small fragment, PCR, amplify the DNA, gel electrophoresis, separate the DNA on the basis of the size, and vectors that carry the DNA into the host cell. So, here correct option is C. The next question is, arrange the following enzyme process in the order of their use for certain. First, you can isolate a DNA. DNA cut by first desired gene. So, this is the desired gene. Then desired gene cut into small fragment by restriction enzyme. Okay. Then desired gene is incorporated into the vector by ligase. Okay. Then the transform okay. The first desired gene, second restriction enzyme, third ligase, fourth transformation, 5 expression. The 5 is 5. Here the A is the correct option. Next question, During insertional inactivation, the presence of a chromogenic substance means beta galactosidase. Beta galactosidase gives blue in color colonies if the plasmid in the bracket does not have an insert. The blue color is produced by the enzyme beta galactosidase. Which of the following is the correct sequence? PCR. For PCR, suppose this is DNA. First, denature at high temperature. So here, denature, then twist and separate. Then use small primer. Suppose 5 prime to 3 prime. So 5 prime to the anil, then primer or anil. Okay. Then 3 prime to 5 prime. So 5 prime, this primer, from this primer, in the presence of Taq polymerase, they synthesis are Taq polymerase and oligonucleotides, in the presence of oligonucleotides or nucleotide, then normal DNA synthesis, that is called anil, and then extension, ultimate 2 DNA strands are produced. So denaturation, annealing and extension is the correct option. 45 number, what does continuous culture means? Continuous culture means continuous supply of fresh medium to the bioreactor to continue culturing the E. coli bacteria or any microorganism. So here DNA and protein getting expressed continuously. Used medium is drained out and the fresh medium is added from another site. This is an option. If there are two type of culture, one is batch culture, the second is continuous culture. In case of batch culture, the nutrition medium given at a time only. But in case of continuous culture, the bioreactor has two opening. From one opening, the fresh medium added to the culture, and another opening, the waste culture media is something given out. Which of the following is a part of biotechnology? In vitro fertilization, synthesis of gene, correcting defective gene, developing of DNA, vaccine. All of these are the part of the biotechnology. Which of the consider the following statement? DNA being hydrophilic. Suppose this is the cell. Okay, this is the cell of E. coli bacteria, E. coli, and this is the recombinant vector. This recombinant or cloning vector is introduced into the E. coli cell for their cloning. So, but this is cell membrane is hydrophobic, and DNA is hydrophilic. So this recombinant DNA do not enter into the host cell easily. So first use calcium divalent ion, then cold and hot treatment. Okay, so DNA being hydrophilic molecule cannot pass through cell membrane because DNA is hydrophilic and cell membrane is hydrophobic. So this is the correct option. Agrobacterium tumefaciens deliver pieces of DNA known as not ZDNA, this is tDNA or Ti plasmid, that is tumor inducing plasmid. Okay, so B is the incorrect option. Retrovirus, adenovirus and papillomavirus are also now used as clone vector in animals, because retrovirus, adenovirus and papillomavirus, all of them are naturally and they infect the human body. So if this vector, if these viruses become disarmed, then it can use as a vector. This is also right one. In genetic engineering, DNA from different sources cut with the same restriction enzyme so that both DNA fragments have same kind of sticky end. Okay, when the different DNA is cut with same restriction enzyme, they produce same sticky end. So right. So here B is the incorrect option. Only B is the incorrect option. Which of the following incorrect about downstream process? What is downstream process? When we produce large amount of product in a bioreactor, after production, the product is collect and they purify and process for marketing. So their process involves separation and purification, right? The product has to be formulated with suitable preservator, also right. Strict quality control testing for each product produced is also required. The downstream processing and quality control testing is the same, not for same. For food product, there is another testing method. For medical product, another testing method. For vaccine, another testing method. So not same product. This is the wrong answer, and this is also correct answer. So here the D is the correct option. Restriction endonuclease enzyme cut the DNA fragment. These fragments are separated by gel electrophoresis, in which negatively charged DNA fragments are forced to move towards the positive. Positive means anode and negative means cathode. Okay, the size of the DNA fragment more it will travel. That is D is the correct answer. 49, 50. Observe the diagram of PBR3. PBR3 means plasmid, Bolivar and Rodriguez. Okay, the 8 restriction site. So 1, 2, 3, 4, 5, 6, 7 and 8. This is right. 4 restriction site have no concern with losing of antibody, because here only BAM H1, PV1, PST1 and SAL1. These 4 antibiotic, 4 restriction site are present in the resistance gene, but HID3, SCAL1, Equaron and PVO2 are not present in the restriction site. So this is the right answer. It has ROP gene. What is ROP gene? ROP gene is a repressor of primer. ROP gene, ROP, R-O-P, repressor of primer that control the copy number of plasmid. Okay, so it has ROP gene. Definitely, copy number means number of plasmid, number of vector present inside the host cell. It has a ROP gene. So here the incorrect statement is none of the. So here this one the correct option. All are correct. Recombinant DNA bearing ampicillin resistance gene is introduced in E. coli. The latter are spread on agar plates containing ampicillin. Then, recombinant DNA bearing in ampicillin resistant. Here, ampicillin resistant genes are present. So, when the E. coli is cultivated in ampicillin containing medium, the E. coli is easily grow in this media because ampicillin resistant genes are present. Transformed recipient cell grow and untransformed cell die. The recombinant no transformed recipient cell grow and untransformed die. This is the correct question. 52. Kary-Mullis. Match the following. The Kary-Mullis discovered the PCR polymerase chain reaction. Kary-Mullis developed polymerase Harvard-Boyer chain reaction. These three. Paul Bartsch, father of the genetic engineering. Stanley Cohen, isolation of antibiotic resistance. A3, B1. So Stanley Cohen said the isolated and antibiotic resistance gene are from plasmid of the bacterium Salmonella typhimurium. And D. Arber, Nathan and Smith, they Nobel Prize for restriction endonuclease. A is the correct option. PCR for DNA amplification. Taq polymerase collect from Thermus aquaticus. Extra chromosomal DNA from plasmid. Ethidium bromide is staining of the DNA part. The A is the correct option. Next question is, start the following statement regarding recombinant DNA technology and select the incorrect ones. Incorrect. The Taq polymerase extends the primer using the nucleotide provided in the reaction. Taq polymer extends the primer using the nucleotide provided. Right. Antibiotic resistant gene are considered as desirable gene. Not antibiotic resistant gene is not a desirable gene because it is used to select the recombinant DNA from non-recombinant DNA. DNA fragments separated according to their charge, not charge, their size. Transformation is a procedure through which a piece of DNA is integrated into the genome of a chromosome. Transformation is a procedure through which a piece of DNA integrated into the host cell. Okay. So, transformation is a procedure through which a piece of DNA is integrated in the genome of a host bacterium. This is right option. To produce higher yield of desired protein, host cells can be multiplied continuous culture. Yes, definitely continuous culture in bioreactor. This is right option. Downstream processing is one of the steps. Downstream processing is one of the steps for collecting the product from bioreactor. So here B, C and F is the incorrect option. Which of the following statement is incorrect? Transformation is a procedure through which a piece of DNA introduced into the host bacterium. Right. Normally, the genes encoding resistance to antibiotics are considered useful as selectable marker. It is also right answer. The normal E. coli cell do not carry resistance against antibiotics such as right. Presence of on recognition sites within the vector will generate several not when the fragment. Suppose this is a bacteria vector. If only one EcoR1 site are present, suppose say for example the only single strand are formed, but when the several restriction site presents, then suppose one fragment, two fragment, it car. So there is, if there is many recognition site within the vector will generate several fragments which will complicate the gene. The incorrect one is D. Next option, how many statements are correct? A thermostable DNA polymerase is used in PCR for repeated amplification. The Taq polymerase. Taq polymerase. Clear? It remains active during high temperature induced duration of double-stranded DNA up to 92 degrees centigrade. The polymerization stage of PCR is where Taq DNA polymerase, which was isolated from thermophilic bacteria Tachymus aquaticus, synthesis the region of DNA between the primers. When double-stranded DNA is denatured by high temp, Taq polymerase become inactive, not false. This is false. So here the A is the, how many statements? A, so 1 is the incorrect. So sorry, 1 is incorrect. So here 3 are the correct option. 56. Actually, he is how many? Correct. The correct option A, B, and C correct option. D only is the incorrect option. So correct option is 3. Next question is, select the correct option to fill out the blanks. Also here agar gel, here a natural polymer extracted from, so here agar gel, agarose gel from red algae but weeds, seaweeds, that is Gelidium, Gracilaria, clear? The DNA fragments purified by gel electrophoresis are used in constructing recombinant DNA by joining them with cloning vector. The ligation of alien DNA is carried out at a restriction site present in one of the two antibiotic resistant gene. Okay, remains active during the high temperature induced denaturation of Taq polymerase. DNA fragments are resolved according to their size through sieving effect in agarose gel. So here A is the correct option. Given below are two statements. Restriction endonuclease recognize the specific sequence to cut a DNA known as palindromic sequence. This is the right option. Restriction endonuclease cut the DNA strand a little away from the center. That is, suppose A, G, C, T, T. Okay, so this is the center portion, but it cut between A portion, T, T, C, G, A. So from the center, this portion. So here both the statement 1 and 2 is correct. The bioreactor is a production of large quantity of product, large tank where the product is produced. Electrophoresis, separation of DNA fragment. PCR is amplification of nucleic acid. ELISA detection of pathogen based on antigen antibody reaction. So here the right option is A2, B1, C4 and D3. This one. Clear? Next is what is primer? Primer is small chemically synthesized oligonucleotides of about 10 to 18 nucleotides that are complementary to the region of template DNA. The primer is a small nucleotide sequence, 10 to 18 nucleotide, and it's complementary to the region of template DNA. Okay, suppose this is DNA and okay, this is template, this is primer. this is primer. Suppose this is 5 prime to 3 prime and 3 prime to 5 prime. Here 5 prime to 3 prime and 5 prime 3 prime. Okay. Then the last question of this chapter is, when restriction enzyme cut the strand of DNA little away from the center, A, G, C, T, T, then stick end, TT, then TT, CG, A, 5 prime to 3 prime and 3 prime to 5 prime. So here the cutting between A, so produce A, G, C, T, T, and here produce T, T, C, G, T, T, C, G, A and A. And this type of ends is called this is called sticky end. So thank you for watching this video. If you like it, please like and subscribe, and if you have any problem, any doubt about any questions, please comment me on comment box. Thank you.